Hello, everyone, and welcome to this AnaBios webinar. My name is Chris Mathis. I'm the Chief Commercial Officer at AnaBios. It's my pleasure to moderate today's session. And before we do that, I'd just like to thank Gary Watkins, AnaBios' Director of Marketing, for putting this together. Today's presentation is the second in a series on pain research. The first, if you missed it, was excellent. Dr. Ted Price spoke on human dorsal root ganglion transcriptomics. If you happen to miss that webinar, you can download it at the link that's shown here. And that link is also posted over in the chat on the right. By the way, there will be questions possible at the end of today's webinar, and you can post them at the chat box, or you can post them in the questions box on the right. And it will also be possible if you raise your hand. We'll try to get to you so you can ask the question live. So the third webinar in our series will be Dr. Laura Stone, and that is next Tuesday, June 23rd, chronic low back pain and epigenetics, perspectives from human and animal studies. So if you haven't done so already, be sure to register for that webinar. So it's my pleasure to introduce our speaker today. Before I do that, I just want to make sure everyone knows about AnaBios. AnaBios is a unique contract research organization and biotech company located in San Diego, California. We recover human tissue samples from ethically consented donors and use the tissue or cells to perform physiological assays in which we can test preclinical compounds for drug discovery research projects. So AnaBios is essentially redefined first in human. So for more information about AnaBios, you can send me an email at cmathis@anabios.com or check out our website. So Cheryl Stucky is the Marvin Wagner Endowed Chair at the Medical College of Wisconsin. She's also Director of the Neuroscience Doctoral Program and Director of the Pain Division of the Neuroscience Research Center at MCW. Cheryl grew up on a wheat farm in central Kansas, received her BA in biology and psychology from Bethel College in Newton, Kansas. She then earned her PhD in neuroscience at the University of Minnesota. After this, she moved to Germany to pursue postdoctoral research, first at the University of Würzburg in Würzburg, Germany, and next at Max Delbruck Center for Molecular Medicine in Berlin. She is currently the lead principal investigator on 3 NIH individual investigator grants from the National Institute of Neurological Disorders and Stroke. One of these is a recent Jacob Javits Award for 7 years of funding. Cheryl's lab studies the molecular, cellular, and physiological mechanisms of sensation, particularly how we sense touch and pain. She is interested in how our skin cells communicate with sensory neurons to convey touch and cold, the causes of severe pain and sickle cell disease, Fabry disease, migraine pain, and the causes of neuropathic and inflammatory pain. She loves training graduate students, postdoctoral fellows, and technicians, and teaching them to write grants, papers, and give talks. So it's my pleasure to introduce Dr. Stucky. Her title of her presentation today is Sickle Cell Disease Pain Mechanisms from Mice to Humans. Dr. Stucky, please take it away.
Thank you very much, Chris, Gary, and Andre for the invitation to present today. So what is sickle cell disease? Sickle cell disease is the most common inherited blood disorder in the United States. 100,000 Americans are affected. That's 1 in 400 African Americans that are affected in the US. And really over more than 3 million people worldwide are affected by sickle cell disease. Now it does affect many people in Central Africa. Up to about 20% of the population in Sub-Saharan Africa can be affected. But it's not just people of African descent that are affected. It also affects people of Indian, Mediterranean, and Middle Eastern descent. So what causes sickle cell disease is a single point mutation in the hemoglobin beta chain, where a valine is substituted for a glutamic acid. So this causes the hemoglobin molecule to form long, stringy polymers when it's deoxygenated. And that causes the red blood cells to become sickle-shaped, and this sickle-shaped red cell then clogs the vasculature, so particularly the small diameter microvessels, and that leads to ischemia in the tissue beyond the vaso occlusion. And the disease is really fraught with a repeated tissue ischemia and reperfusion. In addition, the red cells have a significantly shorter lifespan. It's because the membrane of the red cell is weakened, it lyses and falls apart, and so the red cells have about a tenth the normal lifespan. Normally, red cell lives in your body about 120 days, and these last about 10 to 12 days. So this leads to a chronic hemolytic anemia. So really the patients have chronic microvascular inflammation that is systemic and all over the body. This is artist Hertz Nazaire. Hertz has sickle cell disease, and he creates paintings that depict the struggles that he has with the disease. And his goal really is to inspire empathy and compassion in others to end the suffering that is experienced by the entire sickle cell disease community. A major life-impacting struggle of sickle cell disease is pain, acute pain and chronic pain. And so here, Hertz depicts what sickle cell disease feels, the pain in sickle cell disease feels like for him in his self-portrait, which he has called 10 Redefined. So pain in sickle cell disease. Pain is the most common complication of sickle cell disease. It is the most frequent cause of visits by patients to the emergency department and the most frequent cause of hospitalizations of patients. The pain really comes in two different types. First, there is the acute intermittent pain. This type of pain may correlate with a vaso-occlusive episode that I described when the sickle cells clog the small vessels. So we call those VOEs. And this type of pain typically starts in the first year of life by the time babies are about 5 months of age. And the episodes of these intermittent pain episodes, they increase in frequency and also severity as the patient's age. Now, these are images that I borrowed from my clinical colleague, Amanda Brando, and these images depict a very common phenomenon that occurs in children, which is dactylitis. So dactylitis is an extremely painful swelling of the hands and the feet, and it occurs in pretty much all children that have sickle cell disease. So the second type of pain that occurs with sickle cell disease is chronic pain. Chronic pain occurs not necessarily when there's a vaso-occlusive episode, but it can occur at baseline and be daily. It develops with increasing age. So by the time patients are about 20, many of them suffer from chronic pain, daily pain. So about 40% of adolescents and adults have this type of chronic pain. And Amanda Brando, who is a sickle cell clinician who treats children and adults with sickle cell disease at the Medical College of Wisconsin, has been working with patients to do quantitative sensory testing on the patients and also administering patient pain depict reports to the patient to determine what type of pain the patients have. And she's discovered that many patients describe their pain with neuropathic pain descriptors such as burning, sharp, shooting, stabbing, and prickling. So Amanda is a very close colleague, and one thing that's really wonderful is that as her clinical perspective in treating patients, she sees what the patients experience, and she advises our preclinical studies on a regular basis. So I really like this type of interaction that we have with Amanda, and I find this knowing what she sees in the patients and what the patients experience is absolutely invaluable. So she's also been letting our lab shadow her in the clinic, which has been really wonderful. So patients with more than 3 acute painful episodes per year have a higher mortality or death rate. And overall, the patients, their pain is underreported. It's underreported because only about 6% of the pain that patients experience is ever treated in a hospital care setting. Most of the patients actually suffer from their either acute episodic pain or their chronic pain at home. So this healthcare utilization and the lack of utilization is really the tip of the iceberg of what the patients really experience in terms of chronic pain and acute pain. Now, the pain can be provoked by stimuli. It can be provoked by cold, sorry about that. Pain can be provoked by cold environmental temperatures, by jumping into a cold bath or a cold swimming pool. It can be provoked by even increased wind speed or gentle touch. Now, opioids remain the absolute pain therapeutic that is used most frequently in the clinic, particularly for an acute pain episode. However, many patients are actually refractory to opioid treatment. It does not work that well for the patients. They still suffer pain even when they're treated with opioids. And a big problem is that the patients suffer from discrimination to access adequate health care. Unfortunately, these patients, because of their demographic, they're often considered to be drug seekers when they show up in the emergency department. So what I want to convey to you is that the etiology of sickle pain is quite complex, and it may be both inflammatory and neuropathic. And so my lab has been investigating the pathophysiology underlying sickle cell disease pain. We've been looking at both the inflammatory and the neuropathic basis of sickle pain, and I want to describe to you today a few of the studies that we've done to look at both inflammatory and neuropathic pain. So to look at sickle cell pain, we've been using two preclinical mouse models of sickle cell disease, and I'll describe those here. First of all, we're using the Berkeley mouse model, and what that is that mouse expresses human alpha and beta hemoglobin through an inserted transgene. And the mouse also then has a knockout of the mouse hemoglobin genes. So it expresses, again, only human alpha and beta hemoglobin through a transgene. The second model is called the Townes model. This is a little different in that it is a direct knock-in of the human alpha and beta hemoglobin directly out the mouse hemoglobin locus. Now, when we look at these mice and we look at their pathophysiology, we have found, and others have found, that they recapitulate the patient physiology and pathology extremely well. They have sickled red blood cells, they have chronic anemia, they have significant red blood cell and white blood cell adhesion to the endothelial cell walls. They have vascular congestion and tissue ischemia beyond the congestion, and the mice suffer from severe organ damage. So this type of organ damage can affect all major organs in the body, from the lungs to the liver, to the heart, the spleen, and the kidneys. So we asked, first of all, because we're interested in pain and investigating pain, we wanted to know about pain behavior. So we asked, do the mice have hypersensitivity to external stimuli that would suggest that they may have pain behavior? And in fact, they do. So here are studies that we've done to look at a mechanical and thermal sensitivity on these graphs. A greater sensitivity, more sensitivity will be lower at the bottom of the graph, and less sensitivity will be at the top of the graph. So the mice, first of all, here we looked at mechanical sensitivity by applying von Frey filaments to the bottom of the paw and looking at the von Frey withdrawal thresholds. And the sickle mice are shown in red, controls are in black. you can see that the sickle mice have pronounced mechanical hypersensitivity. And when we looked at cold sensitivity, here we gave the animals the choice to spend time on a cooler plate versus a warmer plate. You can see that the sickle mice spend significantly less time on the cooler plate compared to controls. And we even see this at very small temperature changes like 28 degrees Celsius. So they're very, very sensitive to cold stimuli, and they definitely choose to avoid the cooler side. When we use the Brenner dry ice assay, the sickle animals have a much quicker withdrawal latency when we apply that dry ice stimulus to the bottom of the paw, suggesting, again, acute cold hypersensitivity. And then when we use the Hargreaves paw thermal stimulator to look at heat sensitivity, the mice also show heightened sensitivity to heat. I want to point out that this is all at chronic baseline. So the animals, we do not know when animals are undergoing a spontaneous vaso-occlusive episode. We can induce that experimentally, but all of these data are at chronic baseline. And the pain behaviors begin as early as 7 weeks of age. These particular behaviors were done at 14 to 16 weeks of age. These are all male animals, but we've looked at female animals and they show a very, very similar phenotype. We haven't seen any differences in sexes. Now, the pain behavior gets worse with age, so it's all exacerbated as the animals get beyond 6 months of age. And we can also induce an actual sickling episode, a vaso-occlusive episode, by placing the animals in a hypoxia chamber where we drop the environmental oxygen from normal 21% down to 10%, and we see that particular exposure to hypoxia induces a sickling episode, and that further exacerbates both the mechanical and the thermal hypersensitivity. I just want to mention that the Berkeley and the Towns models have very similar phenotypes with all of these different behaviors. So I will be using those mice interchangeably throughout the data that I'll show. And I also want to highlight that these behaviors that we see in the mice, they correlate very well with what Amanda Brando observes with patient phenotypes. So when she does quantitative sensory testing at baseline, when the patients are not in an acute pain episode, she sees that the patients have increased cold and mechanical sensitivity, and that gets worse as the patients age. And then when the patients are actually in an acute pain episode, that mechanical and cold hypersensitivity also gets worse. So I think it's important to note that the phenotypes that we see in these mice seem to recapitulate very well what happens in the patients. And I really like these models of sickle cell disease because I think that there's an advantage to using them in that the pain behavior develops naturally with the underlying disease. And we know that the disease mimics the disease in human patients, and it's also chronic. So I think it's a fairly natural model of chronic pain that develops from the underlying disease. So because my lab focuses on the peripheral aspects of pain and peripheral drivers of pain, we asked whether the sensory afferents are chronically sensitized in sickle cell disease. So to do this, we took out skin nerve preparations from sickle and control mice. In this case, we're taking the dorsum of the paw that's innervated by the saphenous nerve, and then we place this fresh tissue in a chamber and keep the tissue happy, and we can tease very small filaments from the saphenous nerve until we can get down to recording extracellular action potentials from single units. And then we can stimulate the receptive fields with quantitative mechanical stimuli, cold or heat stimuli, or chemicals. And we can apply antagonists to the receptive field using a mote and then stimulate with, say, mechanical force in the presence of the antagonist. And so these are examples of the action potentials that we get in response to mechanical stimuli. And for this talk, I'll focus mainly on mechanical responses. So we asked whether the sensory afferents are sensitized chronically in sickle cell disease. And here we can show, I'm showing graphs of C fiber type nociceptors and A delta type mechanoreceptor nociceptors. And what you can see is that in response to increasing amounts of force, we see an increasing number of action potentials. And the sickle C fibers that are shown in red here show elevated action potential firing compared to controls to the same forces. So they show nearly a doubling in action potentials elicited by the same force. This is also true for the A delta mechanoreceptors. They show elevated firing. And I'm not showing it here, but even the A beta fibers that innervate Merkel cells and hair follicle receptors show elevated responses. And this may reflect some of the ongoing sensitivity to even light touch. So I want to point out that these nociceptors are chronically sensitized, so we can record as early as 7 weeks of age, and we always see throughout the lifespan that the nociceptors are sensitized. So then we began to ask what might underlie the sensitization to mechanical stimuli. And we had started looking for candidates, and we had seen in some experiments that when we isolate sensory neurons from sickle mice and stimulate them with capsaicin, that they were significantly more responsive to capsaicin. The sickle neurons were extremely sensitive to even very low concentrations of capsaicin at, say, 5 micromolar. So TRPV1 is, of course, the capsaicin receptor, and TRPV1 had been a really promising target in 2010 when we were doing these studies. And TRPV1 had been shown to contribute to the mechanical sensitization in several different types of pain. So even though TRPV1 is, of course, a thermal sensor, it had been shown to contribute to mechanical sensitization in bone cancer pain and several different types of inflammatory pain and neuropathic pain. So we decided to test a potent and selective TRPV1 inhibitor on the sickle cell firing and behavioral responses. And so to do that, we used this compound A425619, which is a very potent TRPV1 inhibitor from Abbott or AbbVie. And what we see is that these are skin nerve recordings here on the left with C fibers. They're all from sickle animals. And when we apply the TRPV1 inhibitor for just 2 minutes in a mote on the receptive field and then mechanically stimulate in the mote in the presence of the inhibitor, again, with just 2 minutes block, you can see that the TRPV1 inhibition, which is shown in blue here, brings that mechanical firing back down to control levels. Then when we inject the TRPV1 inhibitor in an awake behaving animal, we gave it IP here and performed the paw withdrawal thresholds with mechanical stimuli. What you can see here is that the sickle animals are shown in red, and they're down here, they're very, very sensitive to mechanical stimuli. When we inject the inhibitor just one time IP, we largely reverse that mechanical sensitization. And that reversal lasts for about 2 hours. So we've since then tested other TRPV1 inhibitors and seen a very similar effect. We've tested both Berkeley and Towns mice, seen very similar effects in both of these strains of mice. And importantly, we've never seen any effect of the TRPV1 inhibition on mechanical responses in the control fibers or in the control mice. So it seems to be selectively inhibiting this mechanical sensitization in sickle cell disease. However, I'm sure most of you know that targeting TRPV1 clinically, there are a number of problems, and they include that this is the original first generation TRPV1 inhibitors induce pronounced hyperthermia in many patients. That occurs not only in humans, but also in rodents. And then these TRPV1 inhibitors blocked all modalities of activation of the TRPV1 channel, capsaicin activation, proton activation, and heat activation. And then the hyperthermia, there's evidence that the hyperthermia is most likely mediated not by blocking the heat activation of the channel, but rather by blocking the proton activation or a proton-like molecule that activates the TRPV1 inhibitor, or that activates the TRPV1 channel in vivo. And it seems to be occurring in the abdominal wall muscles and the abdominal afferents. So since then, mode-selective TRPV1 inhibitors have been developed that block either the proton or the heat or the capsaicin activation of the channel, so mode-selective activated or inhibitors. But these mode-selective inhibitors have really not had strong analgesic efficacy in clinical trials. So that's unfortunate. And then there's another problem in that blocking TRPV1 peripherally can actually dampen the protective pain in patients against a thermal damage, heat damage. So we asked then, since TRPV1 has so many issues clinically and may not, and we haven't surmounted those problems of targeting TRPV1 clinically yet, we asked, can we target upstream of the TRPV1 channel for sickle cell disease pain? So here, we're looking at inflammatory mechanisms. As I mentioned, there's a chronic pervasive inflammation in patients and animals with sickle cell disease. So can we harness some of these inflammatory mechanisms in order to manipulate TRPV1 activity? And for these studies, I just want to highlight the people that did the work. This is Kate Sadler, who's a senior postdoc in the lab and has been the driver of all these sickle cell studies, as well as other studies in the lab. She's an absolute tour de force and a mover and a shaker. She will be looking for a faculty job in the next couple of years, so I highly encourage you to look out for Kate Sadler. She'll be a fantastic hire. And then the other person that helped drive these studies is Kate Zappia, who's a former MD PhD student in the lab. She's now a resident in Cincinnati. Also just a fantastic driver. So we asked what inflammatory mediators might sensitize sensory neurons in sickle cell disease. So as I mentioned, these patients with sickle cell disease, they're in a tonic pro-inflammatory state, significant microvascular inflammation all over the body. Now, there are a number of pro-inflammatory cytokines that may be involved and are elevated in circulation, but one of those pro-inflammatory cytokines is the chemokine ligand 2, or CCL2. Now, CCL2 it's known to sensitize the TRPV1 channel from prior studies. And it's been shown in sickle cell plasma of patients with sickle cell disease to be elevated. So in this graph here on the right, I'm showing you a study from Quarry and colleagues. These are the levels of CCL2 in the plasma in control, age-matched controls, and then patients with sickle cell anemia during a painful event. So an acute painful event, you can see that their levels are increased by about 20 to 30-fold in the plasma. And even at steady state baseline, when the patients are not in an acute painful episode, they also have elevated circulating CCL2. So CCL2 has been shown to mediate the mechanical hypersensitivity in neuropathic pain models. And so we asked whether chronic inflammation and specifically the CCL2 may sensitize TRPV1 channels in sickle cell disease. And so first of all, we asked, is CCL2 elevated in the plasma of sickle mice, and it is. Now it's elevated, it's not elevated nearly as high, as you'll note, as was true in the patients, but nonetheless, it's still elevated. And the CCL2 ligand will bind to the CCR2 cognate receptor. This is a G protein coupled receptor. And so to investigate this, we used a CCR2 selective antagonist, this RS504393 compound. So we first asked whether the CCR2 inhibition will affect pain behavior. And so for these studies, on the left, here we're testing the mechanical withdrawal thresholds in sickle mice. These are our controls right here. The animals that were injected with vehicle are in white, and the animals injected with a CCR2 antagonist are shown in blue. See, these are sickle traits, so the heterozygous animals right here. But the important thing for this graph is to look in pink, where these are the Berkeley sickle animals. And so we injected the animal sub-Q in the neck with the CCR2 antagonist or vehicle, and you can see that the CCR2 inhibition dose-dependently alleviates the mechanical hypersensitivity, suggesting that it's involved in sensitizing the pain pathways in sickle animals. And then on the right, we injected, here we injected our TRPV1 inhibitor, the A425619, and you can see that in the Berkeley sickle cell animals, the TRPV1 inhibitor dose-dependently alleviates the mechanical hypersensitivity. So then we asked, next, we asked whether CCR2 inhibition is acting upstream of the TRPV1 channel on sensory neurons. So in this case, we isolated dorsal root ganglia from sickle animals or from controls and performed FURA2-based calcium imaging. Whoops, sorry about that. So here you can see the neurons responding to a very low dose of capsaicin. And when we superfuse the neurons with the CCR2 inhibitor in the dish and then test them with capsaicin, what you can see is, first of all, the neurons that are from the Berkeley animals, the sickle neurons, are sensitized to responses to capsaicin. So more of these neurons respond to capsaicin compared to the control neurons. And when we increase the dose of, or the concentration of the CCR2 inhibitor in the dish, we see a reduction of the capsaicin responses in the sickle neurons. So this suggests that the CCR2 activity in the sickle mice and in the sickle neurons contributes to sensitization of the peripheral TRPV1 at the peripheral level. So now I'd like to shift gears here to talk about neuropathic pain. So I'm going to leave inflammatory pain and I'm going to move on to neuropathic pain. We want to know whether there's evidence for neuropathic pain etiology in sickle animals. Is there, first of all, we asked if there is histological evidence for neuropathic pain. And we did this by looking at cross sections of the sciatic nerve from sickle mice and control mice. So here we're looking at the light level at myelinated neurons, and this is a cross section through the sciatic nerve. In control mice, you can see these nice myelinated axons, these thick myelinating axons, which are probably axons of A beta fibers, and these thinner ones are probably the A delta myelinated axons. So when we look at examples of sickle sciatic nerves, what you can see is we saw morphological changes. First of all, we can see with this pink arrow here, an abnormal sort of folding in of the myelin into the axon itself. And we saw quite a few of those abnormal infoldings. Then the other phenotype that we saw is myelin unraveling, and sort of a ring separation of the myelin. And an example is here shown in with this pink star here, where you can see the myelin sort of unraveling. And here are a couple more examples of myelin unraveling. So we quantified this in a blinded fashion. We just quantified the number of times we observed an abnormal infolding or myelin unraveling. When we did that, we saw that in sickle sciatic nerves, that there was a significant increase in the number of pathological fibers with these types of phenotypes. So then we looked at the unmyelinated neurons, and we did this using electron microscopy. And so here you can see in the control sciatic nerves, you can see a nice reed-MAC bundle. So these are the C fibers. And in between are the unmyelinating Schwann cells here. And when we looked at the sickle C fibers, we noted that we found fewer C fibers per reed-MAC bundle. And we also then looked at the blood-nerve barrier by injecting the tail vein in vivo with Evans blue, and then taking out the sciatic nerve and looking at Evans blue extravasation. And here what you can see in the nerve, sickle animals have greater Evans blue leakage in the sciatic nerve compared to control animals. So this suggests that the sickle animals may have a leakier blood-nerve barrier, thereby allowing factors that are in the circulation to access the nerve more easily and sensitize those neurons. In addition, we noted that some of the C fibers actually lost their complete ensheathment by the non-myelinating Schwann cells. So, for example, when we look at a high resolution of these C fibers, in control C fibers, you can see a very nice thick separation by the non-myelinating Schwann cells that sort of individually separates each of the axons. And when we looked at the sickle cell examples, you can see that there's less distance, first of all, between the axons, and there were a number of cases where the Schwann cell appeared to not completely myelinate the axon. So they seem to be losing this fiber insulation around the axon. And we are hypothesizing that this loss of Schwann cell myelination might lead to some cross-excitability between axons, or ephaptic firing in axons. And this is a concept that Marshall Dwar has put forth many years ago, that it is possible that this sort of cross-excitation or ephaptic firing between axons might occur and might potentially be resulting in spontaneous activity in the nerves and potentially contribute to spontaneous pain in the animals. So then we asked, so now that we know there's some evidence of structural anatomical neuropathy, can we repurpose neuropathic pain drugs for sickle cell disease for use in patients? So what about Gabapentin? Now, interestingly, with Amanda Brando, the preclinical, Amanda Brando has looked at patients, and when she administers a pain detect questionnaire to the patients, she finds that 37 to 40% of patients exhibit evidence of neuropathic pain. However, only 5% of these patients have been treated with a drug for neuropathic pain, like gabapentin. And so gabapentin has only been used anecdotally to treat pain in a few patients. So we're asking, is it possible that gabapentin might be an effective therapeutic? So we went back in vivo and tested gabapentin in our animals, in our sickle animals. So in this case, what we did here is we treated the animals daily with a dose of gabapentin for 21 days and then assessed their mechanical sensitivity. So on this graph, on this graph here, these are the town sickle mice that are treated with gabapentin or vehicle. They're down here at the bottom. They're very, very sensitive. And the control mice are up here. And what you can see is even daily treatment for 21 days has absolutely no effect on the von Frey threshold. We also did various other tests like the dynamic brush test, noxious needle test, cold and heat tests, and we found really no effects on chronic administration of gabapentin. There was a very small effect on the needle test, but otherwise no effect of chronic prophylactic treatment on the chronic mechanical or thermal pain. So next we asked whether gabapentin might affect chronic spontaneous pain behavior, because spontaneous is a key complaint of patients with neuropathic pain. So here, we, and I mean Kate Sadler, conducted the condition place preference assay, where after day 1, where she looked at endogenous natural preferences, she then conditioned the animals for 3 days, where she administered vehicle to the animals in the AM and contained them within the box with dots on the sides, and then in the afternoon she administered gabapentin and contained the animals in the chamber with the stripes on the side. And then after 3 days of conditioning, she then gave each animal free rein to choose whether they wanted to spend time on the vehicle or the gabapentin side. And the results are, these are, there we are. These are now the town sickle mice. And you can see that the sickle mice on the test day spent more time, significantly more time in the gabapentin paired chamber than in the vehicle paired chamber. So this suggests that the animals, since they chose the gabapentin chamber, they were receiving some type of ongoing pain relief from the gabapentin. On the other hand, control animals, which I'm not showing here, but the control animals showed absolutely no preference for the gabapentin chamber. So that suggests that gabapentin may have a potentially low abuse potential. So next, we asked whether gabapentin alleviates the pain associated with a vaso-occlusive event. So here, so the past data was with chronic baseline pain, but now we're actually inducing a vaso-occlusive event, and we're doing this experimentally by placing the animals into a chamber where we drop the oxygen content down to 10% for 3 hours, and then raise the oxygen back to 21% normal levels for 1 hour. So for the reperfusion. So all of these animals that I'll show are hypoxia treated. And then we tested von Frey thresholds. So here, what you see is the control mice are here, the town sickle mice are here, and the Berkeley sickle mice are here. And what I want to just explain the legends. So the 1X means the animals went through this vaso-occlusive episode, this hypoxia treatment, and then were tested one time with gabapentin after the hypoxia. The 26X means instead, the animals were treated for 26 days before we put them in the hypoxia chamber. So they were prophylactically treated 26 days before we put them in the hypoxia chamber, and they were not given a post dose of gabapentin. So no post-gabapentin dose here. And then the M indicates that we treated the animals after hypoxia with 1 dose of morphine. And so the important thing to see here is that, so these are the town sickle mice that are treated with vehicle. You can see that they're extremely sensitive to mechanical stimuli compared to control animals. And you can see that 1 dose of gabapentin given after the hypoxia treatment really completely reverses the hypersensitivity that's induced by the vaso-occlusive, the acute hypoxia treatment. And this occurred also in the Berkeley sickle mice. And in fact, this one dose of gabapentin was even more effective at reversing the mechanical hypersensitivity than the one dose of morphine. Also, the one-time dose of gabapentin was more effective than the prophylactic treatment with gabapentin before the hypoxia was induced. So if these findings actually would translate to patients, then it could suggest that when a patient goes to the emergency department for a vaso-occlusive event, that a high dose of gabapentin given right then and there in the emergency department could possibly provide even more pain relief than a high dose of morphine. So we're excited about these data, and it would be really great to test these clinically. So one neural correlate of spontaneous pain is spontaneous afferent firing. So spontaneous firing in afferents. So here we asked whether sickle afferents exhibit ongoing activity after exposure to hypoxia. So here we induced a vaso-occlusive event in the same way that I just described. So we placed the animals in vivo into the hypoxia chamber for 3 hours at 10% oxygen, and then reoxygenated the animals and so reoxygenating the animals, and during the reoxygenation, we then extracted the tibial nerve and placed it into the organ bath and teased the tibial nerve into very, very small bundles so that we could record ongoing activities. So these will be all types of fibers, A beta, A delta, and C fibers, and we're just recording the amount of ongoing activity. What you can see here is that the control neurons that were exposed to hypoxia, they show low levels, very low levels of ongoing action potential firing, but the Townes sickle mice, they often show just a huge barrage of spontaneous action potential firing. They just go crazy, crazy, crazy firing. And again, these are all types of fibers. And when we look at the composite graph, this is the firing frequency, spontaneous firing frequency over time after hypoxia. You can see that the sickle afferent fibers have significantly higher spontaneously firing for between 60 to 80 minutes following that hypoxia. But the control neurons that were exposed to hypoxia show no significant firing. So then we asked, can gabapentin affect this spontaneous firing? Can gabapentin have a peripheral effect on this spontaneous firing? And so what we did here is we superfused gabapentin into the recording chamber after we had induced the hypoxia and after we had taken out these nerves. And what you can see is that the gabapentin superficially superfused into the chamber alleviated this crazy spontaneous firing. And a composite graph here shows that the spontaneous firing treated with vehicle is high. It's about 10 Hertz here. When we just superfuse gabapentin into this chamber, it alleviates, brings that spontaneous firing back down to control levels. So this suggests that the ability of gabapentin to alleviate the vaso-occlusive event pain is due, at least in part, to a peripherally inhibiting this spontaneous action potential firing. So a peripheral effect of the gabapentin on the peripheral afferent axon. So in conclusion, I've shown you that sickle cell pain is mediated by inflammatory pain mechanisms. The TRPV1 channel is sensitized and contributes to this mechanical pain, chronic mechanical pain in sickle cell disease, and the CCL2 binding to its cognate CCR2 receptor can sensitize the TRPV1 channel in sickle cell disease. And then on the other hand, sickle cell pain is also mediated by neuropathic pain mechanisms. I've shown evidence of both myelinated and unmyelinated nerve pathology. And we would like to put forth that neuropathic pain drugs may be effective for treating pain in patients because gabapentin alleviated the spontaneous chronic pain and also peripherally gabapentin alleviated the vaso-occlusive event-induced mechanical pain and the spontaneous afferent firing. So I just want to leave you with some important questions that are not answered, but I think that the field should really think about these questions. One of them is, how do the standard of care treatments for sickle cell disease, those being hydroxyurea and L-glutamine, how do they impact sickle cell disease pain in patients or in these animal models, and how do they impact the development of the chronic pain? So what mechanisms might mediate the transition from the acute vaso-occlusive event type pain to the chronic pain? Is it due perhaps to priming of the pain pathways and the nervous system from the repeated vaso-occlusive crises episodes, because they begin in childhood and they increase in frequency throughout adulthood? Can we identify patients who will develop chronic pain, those 40% that develop chronic pain, from those who do not? Are there biomarkers or genetic markers of those patients that will go on to develop the chronic pain? Is the vaso-occlusive event pain, that acute pain episode, is it really completely explained by vaso-occlusion, or are there other factors that explain that acute pain? And the growing evidence in the clinical field is that it suggests that the vaso-occlusive events do not completely explain the vaso-occlusive or these acute pain episodes that drive the patients to the emergency department. A very important question that's really been untapped largely is, how is sickle cell pain processed by the central nervous system? There are so many directions that can be investigated in the spinal cord and in the central nervous system in the brain. Don Simone has done some beautiful recordings in the spinal cord and shown that spinal cord dorsal horn neurons are massively sensitized to responses. So they're massively sensitized, they exhibit ongoing activity, but very little to nothing has been done in the brainstem or in the CNS or in the brain or in descending control pathways. And then finally, we know that gene therapy is now available for patients, but how realistic is it to treat all patients with gene therapy? And importantly, will this therapeutic actually alleviate their chronic pain? So I just want to leave you with that. And I want to thank the members of my team. In particular, I just want to really call out Kate Sadler, who drove the vast majority of this work. She's a tour de force. She'll be looking for a faculty position soon, and she's just outstanding. And then Kate Zappia also helped drive this work. And then Tony Barand is another postdoc in the lab working on Fabry disease. Alex Meisel is a graduate student in the lab working on the keratinocyte project. Tyler Waltz is an MD PhD student in the lab working on Fabry disease. Tony Menzel is our lab coordinator. Ali Brzezinski is our technician, a Jane of all trades. And I especially want to call out my wonderful clinical collaborator, Amanda Brando, who constantly helps guide us with these preclinical pain models with her amazing experience treating patients with sickle cell disease. And finally, I want to thank my funders from largely the National Institutes of Neurological Disorders and Stroke, and also Advancing a Healthier Wisconsin. And finally, I want to thank you all for listening. I really appreciate it. Thank you for helping us raise the awareness about sickle cell disease pain, because it matters. So thank you very much.
Thank you, Cheryl, for an excellent and very clear presentation. We do have time for questions. Just as a reminder, you can write your questions in the box on the right, or you can raise your hand, and if there's time, we'll get to you and unmute your mic. So I'll get started, Cheryl, as the questions come in. I'm definitely an ion channel guy, so it was very interesting to see the results with gabapentin, which blocks calcium channels and TRPV1. Has your lab gone after other ion channel targets in your research?
No, that's a really great question, Chris. We're interested in looking at sodium channels. For example, NAV 1.7, NAV 1.8. We have not gone after those yet, but I think it's a very interesting question to understand, to investigate how the gabapentin might be alleviating the ongoing firing in the nerve peripherally. And my guess would be it's certainly doing something to dampen down the firing, potentially through sodium channels.
That's interesting. You mentioned sodium channels. And it looks like the mouse models definitely recapitulate the patient pathology, but with rodent models, they haven't always translated well, especially with sodium channel targets like NaV 1.7. We found that with our human DRGs. How do you plan to go after that? I guess, we've talked with your lab about getting human DRGs and doing some collaboration with you, but any other ideas on how to deal with that part of the translational question?
Yeah, I think that's a great question. We've been trying to figure this out. I think it's going to be extremely challenging to get human dorsal ganglia from patients, given the rarity of the disease, the patient demographics, and so forth. However, we're thinking about some ideas with taking, for example, patient plasma. And applying it to human DRG neurons to see what types of activity the plasma elicits, also potentially what kind of gene changes occur when you apply human sickle plasma to human neurons. Of course, those are acute studies. We're interested in potentially generating iPSC cells from sickle stem cells. Those are some ideas we're thinking about.
Very good. All right, so we are getting lots of questions from the audience. Let me start with this one. When measuring nerve activity after mechanical stimuli in the sensory nerve, how do you differentiate C fibers and A delta mechanoreceptor fibers?
Oh, great question. Sure. Well, so normally we're doing conduction velocity when we're looking at mechanical stimuli. So the C fibers conduct much slower. So our cutoff is 1.2 meters per second for C fibers. The A delta fibers conduct between 1.2 and 10 meters per second. Now that, though, is challenging to do when you're looking at such crazy spontaneous activity. So in the spontaneous firing graphs that I showed, we did not distinguish based on conduction velocity. So those were all comers, A beta, A delta, and C fibers.
Okay. And the same person asked, did you see differences in pain behavior using von Frey after VOE-induced mechanical pain?
Yes. In fact, I didn't share the data, but the VOE hypoxia exacerbates their mechanical sensitivity to von Frey and also to stimulation with needle. So it makes it all worse. And that lasts for several hours.
Interesting. What are your thoughts on the mechanism of action for how TRPV1 can contribute to mechanical pain?
That's a fantastic question. My best guess is that it's somehow the TRPV1 channel is somehow sensitized upstream to the bona fide mechanotransducers. Maybe that's piezo2. Maybe it's Takan. We do not know the answer to that, but it does suggest the data from our lab and also a number of other pain models suggest that there's tight coupling between the TRPV1 channel and whatever that mechanotransducer is, because you block that in minutes and that sensitization goes away. I don't know, maybe it's coupling through a lipid mediator. It's a great question.
Next question, I think you addressed in part with the patient population and the difficulty in getting samples from humans. Have nerves from SCD patients been analyzed for abnormal morphology or myelination?
That's a really good question. I don't believe that they have. Yeah, it's difficult to get even those types of samples, but I've been thinking about doing that. So I don't think that they have. One thing that would be easier to get is samples of skin, for example. I know it's a different question, but potentially we could look at keratinocyte responses.
Yep. Okay. Here's another question from the audience. With regard to the increase in firing frequency, for the teased fiber recordings following a vaso-occlusive event in SCD mice. Is it possible that this effect is driven by different types of nerve fibers? And if so, would targeting treatments to those specific types of fibers be an efficient way to treat pain in SCD?
Yeah, it's a good question. I think there are ways we can figure out whether it's A beta, A delta, C fibers, and then I think that it could very well be effective to target channels or mechanisms that are selective for a certain fiber type. So yes, I think we should figure that out, and I think it could be effective, yes.
Here's another question. Are there any other models of SCD pain in other animals, and how do these compare to the mouse models?
Yeah, good question. I thought I heard that I believe that there's a model in pig, and right, and I do not believe that pain behavior has been analyzed in the pig model. So I don't have evidence on that, but there's very few.
Okay. We have more questions. Do you have time to stay on and keep going for a while?
Sure I do.
All right. So at which age do you see significant CCL2 increase in SCD mice, and does VOE increase CCL2 levels compared to non-VOE SCD mice?
Yeah, that's a good question. So those samples were taken when the animals were 3 months old. So they were elevated at 3 months. We did not do a time course to look at when the levels are up, but I think it's a really good question because it would be really interesting to look at the development of the chronic pain and yeah, to look at the time course of the development of chronic pain. And we also did not look at CCL2 levels after a vaso-occlusive crisis, but that's a good idea.
Okay. And a few questions from another member of the audience. How are you planning to look at close proximity cross-firing in the REMAC bundles?
I think that's a really good question, and I don't have an answer for it. But I welcome your thoughts on how to do that.
Well, this person also said, I think it's difficult trying to parse out if it is indeed a cross-influence of firing. I would think it would be based on stimulating one fiber in the REMAC and analyzing a more distant neuron that is unstimulated.
Yeah, okay. That's a fair point. Yeah, that's a good idea to try that. You're right.
And then coming back to the TRPV1 question, do you think it's a physical coupling of TRPV1 to mechanoreceptors or just an overwhelming influx of sodium or calcium leading to gating changes in TRPV1?
Yeah, really good question. I don't think we have evidence of either to parse that out either direction. I was thinking it was a physical, potentially through a tight signaling mechanism, intracellular signaling mechanism or a lipid, but it also could be just the overwhelming influx of positive ions that could sensitize. But then it seems like we should be able to alleviate that and demonstrate that it's either is or is not selective to TRPV1.
Yeah. Okay. And one more question: is the mouse model used typically male or female, or is there a combination? I noticed you had some legend in one of the figures.
Yep, absolutely. So we use both males and females. In the latter part of the talk, all of those studies with Gabapentin were done with half females and half males, and we never have seen a sex difference. And we've done lots of tests with females as well as males, and we've seen very similar phenotypes, very similar responses of the neurons.
Very good. All right, well, thank you very much, Cheryl, and we appreciate your time. Those of you that joined in maybe midway or so, you can download the recording. You'll receive an email with the information. And as I mentioned at the beginning, be sure to register for the next webinar on pain next Tuesday by Dr. Laura Stone. And yeah, thanks you all for attending. Thank you again, Dr. Stucky, and we'll see you later.
Thanks so much, everyone. Thanks so much, everyone, and I'd be happy to entertain questions by email or your thoughts by email. And stay safe, well, healthy, and sane. Thanks again, Chris.
Thank you. Take care.
You too. Bye-bye.